Now we're ready. Welcome everybody in the room and welcome to our online audience for this C-STAR talk. As our country is ravaged by fire, polar vortex and other menacing threats, we celebrate science today with another C-STAR talk. Before I introduce today's speaker, I want to alert you to the talk in 4 weeks' time. We will be joined virtually by Stephanie Forkel from Radboud University. She'll be speaking on a topic that is yet to be disclosed, but I'm sure that's going to be of great interest to our audience as well. Get this out of the way. Here we go. But today, it gives me great pleasure to introduce to you Dr. Samaneh Nemati, who is one of our own. She graduated in 2024, just recently, with Julius Fredrickson in our group here at the University of South Carolina in communication sciences and disorders. Before she joined us here, Samaneh got her master's degree in biomedical engineering from the Ferdowsi University of Mashhad in Iran. And then she joined the world of aphasia and language and cognitive processing. And today she'll not be talking about aphasia, but on a topic that is closely related to a lot of the things that all of us study, including neuroplasticity and associated topics. The title of her talk is On the Relation Between Hearing Loss and Cognitive Decline, and that was also the topic of her dissertation. So, without further ado, Samaneh, it's yours.
Thank you, George, for the introduction, and hello to everyone here in person and also our online audience. I did my dissertation at the University of South, my PhD at the University of South Carolina, as Derek already shared, and today I'm presenting the work that is coming from my dissertation, actually, which I did here. And it is a pleasure to present it as part of the C-STAR lecture series. And before start, actually, I want to recognize that although this work comes from my dissertation, actually it was not my work alone. And I want to recognize the ABC team and all my colleagues and collaborators in the aphasia lab who make this project possible. Being that said, today I'm talking to you about the relationship between hearing loss and cognitive decline in healthy aging. So I will first give a brief introduction and idea behind this study, and then I will introduce the data that we use in this project, which is from the Aging Brain Cohort study, which I will talk about later. And then I will go through the aims of the study, and then at the end I will give a summary and brief discussion. For each aim, I will present the method and results, and then I'll go to the next aim. To begin with, the reason why we think this is important from public health's point of view to study the relationship between hearing loss and cognitive decline is really because of this figure here. This comes from the United Nations, and as you can see, it shows the number of adults older than 60 all over the world. And as you can see, currently it's around 1 billion adults older than 60 in the whole world. And essentially, within less than 30 years, this number will double from 1 billion to 2 billion. And this is remarkable, and we realize why this has happened. Actually, based on some researchers in the field of aging, like Dr. Franklin at Johns Hopkins University, this has happened because we moved to 2 eras of public health in just 100 years. So, in the beginning of 20th century, for the first time in the human history, we began controlling infectious and communicable diseases. And then throughout the 20th century, we began controlling chronic diseases like heart diseases or different types of cancers, which are still the leading killer of human adults, but they are increasingly becoming under control. And so that figure showing the doubling of population from 1 billion to 2 billion is essentially the result of 100 years of public health advances. And now, based on Dr. Franklin, we are entering a third era of public health, and we can name this third era being focused on healthy aging, which is really different from controlling diseases. And now we want to know what are the processes that we can target from a public health standpoint to optimize the health and functioning of an aging population. So there are different areas of research that the researchers are focused now in the field of aging. Obviously, we want to prevent and delay diseases until the very end of life. But of course, if people get diseases, then you want to be able to treat those diseases. So there are a lot of research on effective treatment of different types of diseases to have better aging. And then there are a lot of studies on different lifestyle factors like diet, physical activity, and something like this to understand what is optimal for healthy aging. And then there are some researchers, or a lot of researchers in aging biology who studies the underlying biological processes with aging. And with those research, for example, there are some hopes that in the future we may have some drugs that can target specific or certain systems in the body, like, for example, reducing muscle loss or optimizing heart function, for example. And then this last item that I have listed here, which is optimizing sensory function, is a little bit stuck on, and there's not a lot of research on that. One reason could be that there is not enough evidence about what are the consequences of sensory loss in aging to then think about how to optimize sensory loss. And of course, all senses are important, and all sensory loss has consequences for aging population, but here our focus is on hearing for several reasons. And one reason is this figure, actually. So this shows prevalence of hearing loss in the United States, but by each decade of life. And you can see in early ages, it's just a couple of percent, but then this prevalence of hearing loss is almost doubling by each decade of life after a certain age, like around 40 or so. And if we look at the people who are 70 and older, almost 2/3 of them will have a meaningful hearing loss. So it makes sense that 100 years ago, from public health standpoint, it wasn't actually of much concern to look at sensory loss because we didn't have a lot of people who age that much. But now with this increasing aging population, we have a lot of people who age 60, 70, and 80. And actually, it's not the matter of if they lose part of their hearing, but when they lose part of their hearing at some point. So hearing has become an increasingly important determinant when we think about how people can age in a successful and healthy way. And also, this is important because not just hearing or hearing loss affect people's communication ability. It's also important because there are some evidence that hearing loss is a leading factor to other health conditions. For example, Lin et al. In 2011 have shown that people with mild to moderate presbycusis, which is another word for age-related hearing loss, show worse performance on executive function tests. Or, for example, some other studies have shown that hearing loss is associated with global cognitive decline and social isolation and also depression. And in line with this evidence, Lancet Commission on Dementia convened in 2017 to review best available evidence on dementia and to come up with some recommendations on how to manage or even prevent dementia. And this infographic here comes from the report in 2024, and it shows percentage reduction in cases of dementia when each of these risk factors is controlled or prevented. And if I zoom in, you can see that what they reported was that hearing loss has the highest proportion of risk for dementia among other factors, and it's 7% of dementia cases globally. So this means that 7% of dementia cases globally could potentially be prevented if hearing loss is prevented or treated. But one of the assumptions of this study is that each of these risk factors, like hearing loss, lead to dementia, which is something that we don't know yet, actually. But there are some hypotheses or mechanisms in the literature through which researchers think that hearing loss can lead to cognitive decline. One of these hypotheses is information degradation, which is also referred to as cognitive load. So it's like hearing has two steps. One step is happening in our ear, but the second step happening in our brain. So the sound comes to our ear, and then it is sent to brain for further processing to understand the information. And if, for example, we have some problem in our ears, let's say in our cochlea, then the processing in the ear doesn't happen correctly, so the encoding doesn't happen correctly, and the signal that is sent to the brain is not a very clear signal. It's kind of degraded or garbled signal. And in that case, the brain needs to put much more effort to understand that information and decode the information, actually. And that extra effort that the brain puts to understand the sound and decode information comes at the expense of not being able to do other things, like, for example, a memory task or remembering things. And it might be one of the reasons that it's harder for older adults to, for example, hear a phone number and repeat it after you or write it down. And then there is another hypothesis, sensory deprivation, which is about the direct effect of hearing loss on brain. And it is actually, it says that because of lack of input to part of the brain that is responsible for hearing, people will lose that part of the brain gradually, because brain is like your muscle. If you don't use it, then you lose it. And actually, the effect is not limited only to auditory areas, because as we know, the brain works as an interconnected network, so damage to auditory-related areas can then lead to damage to other parts of the brain or damage the function of other parts of the brain. So the sensory deprivation hypothesis starts with damage to hearing, processing parts of the area, but then it extends to non-auditory areas as well. Another hypothesis is social isolation. So as we said earlier, hearing loss affects people's ability to communicate with others, and because of that, actually people with hearing loss are less inclined to have interaction with other people and do social activities, and this may affect their cognition in the long run. And apart from this causal hypothesis, there is this non-causal hypothesis, which is called common cause. And it is an important one because it says that there might be a common factor which leads to both hearing loss and cognitive decline at the same time. And these common factors could be inflammation or vascular disease, which is actually prevalent with older adults. And so this common cause can lead to both hearing loss and cognitive decline, instead of hearing loss leads to cognitive decline. But it is most likely, at least based on some researchers, that these hypothesis pathways coexist together and leading to cognitive decline rather than one of them being true. So this is the context that provides motivation for this study to increase the understanding of the relationship between hearing loss and cognition in healthy aging. And I should note that we are not looking at causal relationship here actually, because our data is a cross-sectional data and not longitudinal. But the aims of our study was to first identify specific changes in the brain structure, specifically in the gray matter volume of the brain, and check if these changes are actually kind of mediating the relationship between hearing loss and cognitive decline in healthy participants, in healthy adults. And then the second aim was to investigate the mediating role of regional brain age gap in the relationship between hearing loss and cognitive decline. And then the third aim was to identify the mediating role of hippocampus, anterior cingulate cortex, and frontal regions in the relationship between age-related hearing loss and specific cognitive domains like memory, executive function, and attention. And these specific domains were chosen because they are known to decline prior to dementia onset based on the literature. And the data we used in this study comes from the Aging Brain Cohort project at the University of South Carolina. It is an ongoing project, but we included 231 participants in this study, and they aged between 20 to 80 years old. So they were all participants who had completed all the measures we needed for the objectives of this study when we were doing this. So there are a lot of measures collected at the ABC study, but we use measures that were in line with our objectives actually. So we use demographic information like age, sex, and so on. And we use their T1 weighted MRI scans to calculate gray matter volume and the brain age. And then for cognitive assessment, we use scores from MoCA test, which provides a MoCA total score, which is indicative of general cognitive performance of participants. And also it provides some sub-scores, including memory, executive, and attention. So for the aims 1 and 2, we use MoCA total score. And then for the aim 3, when we look at specific cognitive domains, we use sub-scores of MoCA test. And for hearing assessment, we did 2 tests, pure tone thresholds and words in noise test, which is part of the NIH toolbox. So in PTT test, tones in different frequencies ranging from 0.25 to 8 kilohertz were presented to participants, and they were asked to signal when they detect the tones. And in word in noise detection test, participants were presented with spoken words with increasing levels of background noise, which is kind of simulating real-world situations like when you are in a restaurant or any crowded places and you need to understand speech among other noise or sounds, or understand speech from a specific person when multiple people are talking. So here you see the pure tone threshold for older versus young participants. Older adults are shown in red, and young adults are shown in blue. And by older, I mean people who age 60 and above. And the top figure is for left ear, the bottom figure is for right ear. And if you look at the red line, you can see that it has an upward trend, which shows that with higher frequencies, older people actually has higher pure tone thresholds. So the tone or the sound needs to be louder for them to hear that. So higher threshold or higher PTT means worse performance on hearing tests. And for PTT scores, actually we calculated better ear pure tone threshold averages. So whenever in this presentation I say PTT score, I mean better ear pure tone threshold averages. And here you see PTT scores and WIN scores versus age. So PTT is shown in red and WIN is shown in blue, and age is the x-axis. And again, as you can see, as age increases, PTT score and WIN score increase, which again indicates that older adults actually have higher PTT and higher WIN scores, which shows worse performance on pure tone threshold and words in noise test. And then here you see the relationship between all behavioral scores that we use in this study. The first one, the left figure, is for PTT versus WIN score, which shows significant positive correlation, and then we have significant negative correlation between PTT and MoCA total scores, which makes sense because worse performance on PTT test is associated with lower performance on MoCA test. And then the same for Venus score and MoCA total. And here is the range and mean for each of these variables. One more thing before I go to the aims and explain the aims of the study is about how we parcellated the brain. So we used JHU atlas to parcellate the brain, but then we replaced superior temporal gyrus with the one from another atlas, which has been developed by Connectivity Toolbox. And it is a combination of AAL atlas and Harvard-Oxford atlas. The reason for that was it was important for us to have a fine grain parcellation in superior temporal gyrus because we are looking at hearing. And this atlas provides divisions for Heschl's gyrus, planum temporal, and posterior superior temporal gyrus, which were important regions for us. And so totally we had 195 ROIs, but then to reduce the number of ROIs to a reasonable number actually, we chose certain ROIs, and these were chosen based on the literature, which includes auditory areas and also non-auditory areas, which are actually involved in broader cognitive processing, which have interaction with auditory processing based on the literature. And then we group those ROIs to create these large brain regions. So as you can see, we have hearing-specific regions, which include Heschl's gyrus, planum temporal, and posterior superior temporal gyrus. Then we have domain general, which includes frontoparietal regions, and then language-specific region, which includes regions from frontal and temporal lobe, and then cingulo-opercular network, which includes anterior cingulate cortex, insula, and thalamus. And here you see those regions on the brain surface. So the first aim of the study was to identify specific structural changes in the brain through which age-related hearing loss might be associated with cognitive decline. And we did 4 steps of analysis to achieve this aim. So we extracted gray matter volume of each ROI based on T1 weighted MRI scans of participants using CAT12 toolbox in SPM in MATLAB. And then we did simple correlation analysis, again in MATLAB, to see the correlation between gray matter volume in each ROI and hearing score. This was great and gave us a good idea about the relationship between what's going on in the brain and the hearing scores, but actually this couldn't provide a good understanding about non-linear relationship or potential interaction between variables. And also we wanted to know what parts of the brain are more important about predicting hearing scores. So we use random forest regression, which consider potential nonlinear relationships and interactions between those variables and hearing scores, and also it provides us with feature importance to show what parts of the brain are more important in predicting behavioral scores or hearing scores. And we use scikit-learn package in Python to do this. We use 100 trees and 49 predictors, which were 48 ROIs plus age actually. And then finally we did mediation analysis to see the direct and indirect effect of gray matter volume in these ROIs in the relationship between hearing loss and cognitive decline. Because you will hear mediation analysis from me a lot in this presentation, especially when I go to the results, I thought it would be good to explain this a little bit. So we had 2 mediation models actually. The first one was for PTT score, and so the independent variable in the first model was PTT score, and the dependent variable was MoCA score. And then we use gray matter volume in brain ROIs as mediators. And those mediators were chosen based on the results we had from correlation analysis. So we chose only significant regions as mediators. And then we had a similar model for Wien scores. And there are some terms with mediation analysis to explain. So there is total effect, which is the effect of independent variable on the dependent variable without considering any of the mediators. So it's like a simple correlation analysis. And then we have direct effect, which is the effect of independent variable on dependent variable while accounting for all mediators in the model. And then we have indirect effect, a total indirect effect actually, which is the effect through all the mediators at the same time. And then we have a specific indirect effect, which is the effect through one specific mediator. So let's say, for example, in this model, we will include 3 moderators. And then when we say there is a significant total indirect effect, it means that we have an indirect effect through all 3 mediators, which mediate the relationship between PTT and MoCA. And when we say there is a significant, specific indirect effect of M1, it means that indirect effect through only one mediator, M1. So here you see the results of the correlation analysis we did between gray matter volume in brain ROIs and the hearing scores. And these are regions which showed significant negative correlation with both PTT and Wien. So from auditory cortex, we have Heschl gyrus and posterior superior temporal gyrus in both left and right hemisphere. From domain general region, we have orbitally section of inferior frontal gyrus in both hemispheres. And then from cingulo-opercular network, we have insula from both hemispheres, which showed significant negative correlation with both PTT and Wien, which means that higher PTT scores or higher Wien scores were associated with lower gray matter volume in these regions, which is like worse hearing is associated with lower gray matter volume. And here are the scatter plots of those relationships. So the left is auditory areas and the right is non-auditory areas. And red shows PTT and blue shows Wien. And as you can see, there is a negative relationship there. And then these regions are the ones which showed significant negative correlation only with Wien score, but not with PTT scores. And from domain general regions, we have superior frontal gyrus, middle frontal gyrus, specifically dorsolateral prefrontal cortex, and posterior cingulate cortex in both hemispheres. And from cingulo-opercular network, we have rostral anterior cingulate cortex in the right hemisphere. And these are, again, the scatter plots of those relationships. And here is the results of random forest regression for PTT score. So here you see the model metrics, and when predicting PTT score, we also included age as a predictor. And you see that the R-square of the model is pretty high, it's 0.69, which means that actually the model could predict around 70% of variance in PTT scores. And also we have mean square error and mean absolute error. So the mean absolute error shows that the model predicted scores were within 4.39 units of the actual score of PTT. And then because the age was included in the model and we were thinking maybe most of the predictive power of the model comes from age and not the brain ROIs, but we did the model one more time without including age actually as predictors. But as you can see, the model performance changes a little bit, not dramatically. So the R-square or the model performance is still pretty high, it's around 65%. So this shows that age does contribute to the predictive power of the model, but it's not significantly dominant. And a gray matter volume in those brain ROIs on their own still accounts for the substantial portion of variance in PTT scores. Then here is the results for random forest regression for Venus scores. Sorry, it's still PTT scores, but it shows the feature importance. So if you remember from correlation analysis, we had a lot of regions that showed significant negative correlation, but then we wanted to know what parts of the brain are more important in predicting PTT scores. And this bar here shows the higher bar shows that those regions are more important in predicting PTT scores. And the first one is right insula, which is the most important one in predicting PTT scores, and then right Heschl's gyrus, and then age and others. And as you can see again, age is still important, but not the most important factor in predicting PTT scores. And here are the results for Venus scores. So the model performance is not as good as the PTT scores, and it's much lower. It's around 40%. But again, when we exclude age and repeated the model metrics actually don't change a lot, and it's just minimal changes, which again shows the model's prediction of Venus score is largely independent of age and relies primarily on the gray matter volume of those ROIs, but in general it's much lower than predicting PTT scores. And the feature importance for Venus scores is interesting. Still, right insula is the most important factor to predict Venus scores, but and then it comes anterior cingulate cortex, IFG, MFG, and so on. But one important thing is that although right insula is still the most important factor to predict Venus scores, feature importance for Venus scores is more evenly spread across multiple ROIs compared to PT scores. And this may reflect the complexity of speech processing compared to like just sound processing, like tone processing in PTT, because it needs a broader network in the brain to process speech rather than just tones. And here is the results of mediation analysis for PTT scores. In this figure, wherever you see values, it means that there was a significant relationship in that specific path. And when you see red cross, it means that there wasn't significant relationship on that path. There is a lot to explain about this figure, but for the sake of time, I would focus on the indirect effect, which is more interesting and important to us. So here the PTT is the independent variable, MoCA is the dependent variable, and then we wanted to see if gray matter volume in those specific regions which showed highly significant correlation actually with PTT scores are kind of mediating the relationship between hearing and cognition. And so mediators are gray matter volume in Heschl's gyrus in left and right hemisphere, and also gray matter volume in insula in both left and right hemisphere. And that actually arrow at the top shows the total or combined indirect effect of those mediators. And as you can see, there was a significant total indirect effect through all those four mediators. And when we calculated the proportion mediation effect, so we divided the strength of indirect effect to total effect, and it shows that it's around 59%, which means that actually 59% of the relationship between PTT and MoCA is mediated through gray matter volume in those specific brain regions. But there was no significant specific indirect effect through a single ROI. And then in the second model for Venus score, we have kind of similar results with this difference that mediators for the second model were more But for representation, we just show 4 of them because otherwise we had a very messy figure. But here again, we have a significant total indirect effect through all those mediators, but we don't have any significant specific indirect effect through a single ROI. And also for Venus score, actually the mediation effect is not as big as PTT scores. It's around 25%, which means that 25% of the relationship between Wien and MoCA is mediated through gray matter volume in those brain regions. And because it's not a very high proportion mediation effect, actually, it might reflect that there are some other mediators that is mediating this relationship that are not included in this model. And then based on the results that we have from random forest regression or feature importance analysis, we had the right insula as the most important one. So we created the mediation models one more time with including only right insula as mediator, as a single mediator. And again, 59% of the relationship for PTT and MoCA was explained by gray matter volume in right insula, and for Venus score, actually, it was around 22%. And then for the aim2, we...
I think you can click on that X here. I think.
For the aim2, we wanted to investigate the mediating role of regional brain age gap in the relationship between hearing loss and cognitive decline. And so for this one, first we calculated brain age gap for those large specific brain regions that I showed you in that table at the beginning of the talk. So like hearing specific region, domain general region, cingulo-opercular, and language specific regions. For the actually total brain age, we use Brain Ager toolbox from GitHub from James Cole to calculate that. But for regional brain age gap, we use the method from Busby et al. Paper in 2024. And by brain age gap, we mean actually the subtraction of chronological age and the estimated brain age. So I will explain this in the next slide a little bit more. And then similar to the previous aim, we did the first correlation analysis to see the relationship between brain age gap in those regions and the hearing scores, and then mediation analysis to see the total direct and indirect effect of brain age gap in mediating the relationship between hearing and cognition. So for brain age gap, actually we created a linear regression model using gray matter volumes of brain ROIs as predictors and chronological age of participant as outcome variable. And then each time, actually, when we wanted to create that model, each time we excluded one subject and considered that as a pseudo patient and didn't include that in that model, in that linear model actually that we created. And then when we wanted to calculate the brain age for this participant, we use the gray matter volume of all ROIs within a specific region. Let's say we wanted to calculate brain age gap for hearing specific regions. So we use gray matter volumes of all regions within these large brain regions like Heschl gyrus, posterior superior temporal gyrus, and planum temporal as predictors, and then we calculated their brain age. We did this for all those large brain regions, domain general, cingulo-opercular, and language specific. So after we had the brain age for each of those regions, then we subtract the chronological age from estimated brain age to calculate brain age gap for each specific regions for each subject. So here is the results of the correlation analysis for brain age gap. And these regions are the ones that showed from hearing specific actually region which showed significant positive correlation with both PTT and VIN scores, which shows that higher brain age gap or accelerated brain aging is associated with higher PTT and VIN scores or worse hearing. And then for domain general regions, again in both left and right hemisphere, we had significant positive correlation only with the VIN scores, but not PTT scores. But again, accelerated aging in domain general region was associated with higher VIN scores, which showed worse performance on words in noise detection test. And then the right cingulo-opercular region had actually kind of a borderline significant, not really significant, with VIN scores and not with PTT scores. And here are the scatter plots for those relationships for hearing specific regions, domain general, and cingulo-opercular. And then the mediation analysis for this one is similar to the previous aim, with this difference that this time mediators are brain age gap in specific brain regions rather than simply gray matter volume. So again, the independent variable is PTT score, the dependent variable is MoCA, and then the mediators are brain age gap in hearing specific in left hemisphere and hearing specific regions in the right hemisphere. And again, where you see values, it means that there was a significant relationship in that specific path, and when you see red cross, it means that there was no significant relationship in that path. So as you can see, we didn't have a significant direct effect from PTT to MoCA, but we had significant total effect and also a significant combined indirect effect of hearing specific regions in left and right hemisphere, and also a significant indirect effect through only hearing specific regions in the right hemisphere. So the proportion mediation effect for the combined effect of hearing specific regions in both left and right hemisphere was around 64%, which shows that 64% of the relationship between PTT and MoCA scores was mediated through accelerated aging or higher brain age gap in hearing specific regions in both left and right hemisphere. And then we also calculated proportion mediation effect for only hearing specific region in the right hemisphere, because that one also was significant. And it shows that around 71% of the relationship between hearing and MoCA, specifically between PTT and MoCA, was mediated through accelerated aging in hearing specific regions in the right hemisphere. We had similar results for VIN scores, with this difference that both total effect and direct effect here are significant. So we have significant total effect from VIN to MoCA, and also significant direct effect of VIN on MoCA. And then the indirect effect, the total indirect effect through both left and right hearing specific regions were around 22%. So 22% of the relationship between VIN and MoCA was mediated through accelerated aging in hearing specific regions in left and right hemisphere. And again, because this number is not very high, it might reflect that there are some other mediators that are mediating this relationship that are not included in this model. Or it could be that the relationship between VIN and MoCA is more direct actually rather than being mediated by other factors. And then for the third aim, actually the third aim had 3 sections itself. So in the first section, we wanted to see the relationship between hearing loss and memory, and also the mediating role of hippocampus in this relationship, because hippocampus is known to be involved in memory processing. So we did a correlation analysis, again, between hearing scores and sub-scores like memory index scores of MoCA, and then correlations between hearing scores and gray matter volume in the hippocampus. And then we did a mediation analysis to see if gray matter volume in hippocampus mediates the relationship between PTT and MoCA index scores, MoCA memory index scores. And so for the sake of time, I just skipped the results from correlation because we can see them in the mediation model as well. And here is the model for PTT and the memory index score. So you can see that we have significant total effect, significant direct effect, but we don't have any significant indirect effect. So the relationship between PTT and MoCA index score is not significantly mediated through hippocampus, actually, as we had hypothesized. And for Win scores, we have significant total effect, significant direct effect. We don't have significant total indirect effect when we include both hippocampus in right and left hemisphere, but we have a specific indirect effect of right hippocampus. So when we consider the total effect of hippocampus in both right and left, we don't have any indirect effect, but considering only hippocampus in the right hemisphere, it is mediating the relationship between Win and MoCA memory index score. And so again, we calculated the proportion mediation effect by dividing indirect effect to total effect, and it seems that around 48% of the relationship between Win and MoCA memory index is mediated through gray matter volume in the right hippocampus. And then in the second section of the AIM-3, we wanted to see the relationship between hearing loss, attention, and the mediating role of anterior cingulate cortex, because it's involved in attention based on the literature, and we chose that region because of that. Again, we did correlations between hearing scores and MoCA attention index score, and then correlations between hearing scores and gray matter volume in anterior cingulate cortex. And then we did a mediation, we created a mediation model similar to before, with this difference that mediator here is the gray matter volume in anterior cingulate cortex. And based on the atlas that we had used for parcellating the brain, anterior cingulate cortex has these subdivisions in JHU atlas: rostral, dorsal, subcallosal, and subgenual. So we calculated the average gray matter volume of these regions to include as mediator in the model. And so here's the results for the mediation model for PTT and attention index score. So as you can see, there was no significant indirect effect, and also no significant indirect effect through anterior cingulate cortex between Win and attention score. And then in the last section, we wanted to see the relationship between hearing loss, executive function, and the mediating role of frontal lobe in this relationship, because frontal lobe is known to be involved in most of executive function processing. So this analysis was similar to the previous section. We first did separate correlation analysis between the behavioral scores and also between gray matter volume in frontal lobe and the hearing scores. And frontal lobe included, the regions that we included actually was superior frontal gyrus, middle frontal gyrus, IFG and middle frontal orbital gyrus, gyrus rectus, and pericentral gyrus, and it was again based on the parcellation that we had with those two atlas, or the JHU atlas. And so we calculated the average gray matter volume in these regions, and then we included this as the mediator in the model. Here is the results for PTT and executive function score. So as you can see that there was a total indirect effect, a significant total indirect effect through frontal lobe in right and left hemisphere, but there was no significant specific indirect effect. And for Wien scores, again, we have a significant total indirect effect, which means that the relationship between Wien and executive function was mediated with gray matter volume in frontal lobe in both left and right hemisphere, and it was around 26% for the Wien score. So from the AIM-1, we saw that we had the reduced gray matter volume between both auditory and non-auditory ROI were associated with poorer hearing scores in both PTT and Wien, and this can be explained through two frameworks. Actually, we can say that there is a common factor that leading to both of these changes, like changes in hearing processing and also changes in brain. But there is also this hypothesis, as I showed in the introduction, that there could be a causal relationship that, for example, changes in the brain, like, for example, because of lack of input to the brain, that part of the brain that is responsible for hearing processing is changed or is damaged, and then this change lead to worse score or worse performance. And then we had right insula as the most important factor, actually, to predict PTT and words in noise processing, and right insula is part of the cingulo-opercular network, which is based on the literature, is a strong candidate network beyond the auditory areas that people use in response to degraded signals, either in the case of, for example, exposing to the background noise or having hearing problems. And then we saw that accelerated brain aging in hearing-specific regions were associated with higher PTT and Wien scores, which means that accelerated aging in hearing-specific regions, including Heschl's gyrus, planum temporal and posterior superior temporal gyrus, were associated with worse performance in PTT tests and worse in noise detection tests. And we had the mediating role of specifically right hearing-specific regions in that relationship. And then we also saw accelerated brain aging in domain general regions, which were associated with poorer Wien performance or worse in noise performance, but we didn't see any mediating role in that relationship. And then we also saw mediating role of right hippocampus in the relationship between words in noise detection and memory performance, but not for PTT. And then at the last part, we also saw mediating role of frontal regions in the relationship between hearing and executive function. And so, as I said at the beginning of this work, we couldn't see the causal relationship here because actually the data we had was cross-sectional. So it would be great that in the future we have longitudinal study to be able to use longitudinal data to verify whether changes in hearing loss precipitate changes in the gray matter volume and cognitive decline. And it is important to understand for a couple of reasons, but most importantly because actually we need to have a causal relationship between hearing loss and cognitive decline to then think about, having an intervention on hearing loss and be able to delay cognitive decline and potentially later dementia in life. And then so I think after we can confirm that the relationship is causal, then we can think about intervention studies to intervene on hearing loss. So I wanted to thank actually everyone who helped me with this project, especially the ABC team, Roger and Sarah Newman, Norland, and everyone in the aphasia lab, actually, including speech language pathologists, researchers, postdocs, students, and all the faculties who collaborated with us, and also my advisor to make this project possible. And last but not least, the participants of the study who made this possible. And I'm ready for your question.
Thank you so much, Samaneh.
What a great body of work. Let's start with questions from our audience.
Really. So, Samaneh, I love that you chose these two tasks in particular because I think one is really pure auditory perception and one is so much more complex, but also relies on auditory perception. And I think of the words and noise almost like, or to think of like in my daily life, I have teenagers and they love to mumble, and thank goodness I actually can fill in the blanks for what they're saying and still get what they're saying, because I would sound like this, but I still can understand them. And what I like about this task is that for the words and noise, when it becomes really difficult to hear, because it's actually difficult for even people with healthy hearing to start to hear it when the noise ratio gets too high, that it's almost kind of introducing this, you have to fill in the blanks for what you heard versus what you didn't hear for the words. And so what I like about it is that it's kind of, you have to use this cognitive part, right, to actually execute that task. And I was wondering if you could maybe like speak a little bit about two things. One is, like, are there people who do well on PTT who don't do well on words and noise, that are kind of have more of a cognitive only deficit? Are there people who, you know, I saw that did poorly on both, but also those that just do poorly on PTT and not? To kind of separate these mechanisms where you should see kind of the more vast degradation or atrophy of the brain when they have both versus one versus the other, where you might actually still have intact gray matter volumes, for example.
That's a great point and a great question, actually. But I have looked at if people do, for example, bad in PTT, but not in words and noise, but not the other way around. So it's a good way to do that. I mean, when I looked at people who did bad on PTT, but they were still doing good on words and noise, there were just a few numbers, so I didn't separate them in further analysis. And I think if we have more people, then we can do that. But as you said, it would be also great to look at those who didn't do good on word, but did good on PTT. I haven't looked at that.
Questions from the room?
Samaneh, I don't see an online question yet, but I had one that I entered earlier. Just if you had to choose an intervention for age-related cognitive decline, under the assumption that you want to intervene, right? Because it's also just normal age-related cognitive decline. Would you want to target hearing loss, or would you work on brain health, or which one do you think is easier to target, for example? Which one would be like more clinically feasible or more effective? Do you have any thoughts about that? Or?
Well, I haven't thought deeply about that, but I think the most important thing first is to confirm that which one is leading to which one, actually. I mean, it could be both, right? It could be cognitive decline leads to like worse hearing, or it could be worse hearing leads to cognitive decline, or neither of these. And there is a third factor that is leading to both. So I think the most important thing is to first confirm that which one is leading to both, to what, and then think about which one to intervene.
In the models that you presented, you basically started off with the hearing loss, right? You were
Yes.
looking at whether the hearing loss mediated by something else had an effect.
Yes,
But that's not necessarily your
that's
ground.
not necessary, but we did this because our hypothesis actually were shaped based on this hypothesis in the literature that like the sensory deprivation hypothesis that says, for example, because of lack of input to hearing part of the brain, we have some, difficulties in hearing, and then this leads to other changes in other parts of the brain, which then leads to cognitive decline. So we went based on that hypothesis, but of course there are other hypotheses in the literature that can be tested. And I think before thinking about which one is easier or better to intervene on, it would be better to first confirm which one is leading to which one.
Great job, Samaneh. I wanted to see if maybe you could dive into the longitudinal data for ABC and see, because initially when we tested all those adults that were 60 and above, most of them did have difficulty with the words and noise test, and then also the thresholds. Practically all of them, did not do well with the higher frequencies, which is pretty common. Not everyone had corrected hearing at that time point. A few did, if I remember correctly. But I'm curious, in that time period, the 5 years, do they now, did they go see an audiologist? Did they have their hearing tested? Do they have hearing aids? Is it corrected now? Are they performing differently? That might be interesting to look at over time.
Yes, it would be great. I don't know how many longitudinal data we have right now, but I know probably 40. That's great, yeah. Because when I was doing the analysis, we didn't have quite that number actually, so we didn't do longitudinal analysis. But now that we have more data, I think, yeah, that's great to do that. And look, for example, if those with hearing loss got worse, and also if, for example, changes in the brain are worse or no, and it would be great to look at that.
We have so many social emotional surveys and stuff like that, because it would have a huge impact. If they're having difficulty hearing out in the community, sometimes they just retreat back and start to not do those things, or not communicating with others as much. So it'd be interesting to look at some of our measures. Yeah, that's cool.
We have a suggestion from Margaret Pichura Fuller on a paper, and I've copied that reference, so I will leave that for you. It's Ban Singh and Pichura Fuller 2012, Age Effects Responses on the Speech, Spatial and Qualities of Hearing Scale for Adults. So if you're not aware of that paper,
Thank you.
you are. And there's a question from Portia Washington. What are your thoughts about interpreting brain age versus the brain age gap as described here, or other indicators of structural brain health? And I've asked her to expand on that question, but maybe, she says, yes, some programs I've seen spit out an actual brain age similar to chronological age, and the gap is between the two.
Yeah, actually, if we look at the brain age itself, it's highly correlated with the chronological age in our sample, at least. And so we didn't look at the estimated brain age, and also similar to the other studies in the literature, which looks at the difference between brain age and chronological age to see if the brain is actually accelerating in aging or decelerating in aging. We cannot know that from just the brain age itself. With brain age, we can just know that the age of the brain is this, but with the gap, we can know if it is accelerated brain aging or decelerated.
I would assume, just as an aside, if you would in your models, if you would replace chronological age with brain age, things wouldn't change a lot, right?
Yes.
But it's the gap that makes that gives the extra information. Yeah.
Great. It's possible I can ask a follow-up. Why is the gap important? I know we've talked about this in various times, but if I'm thinking about an animal and it's like, they're prematurely aged, right? And the problem is their body is older, right? The fact that it's older is the problem that you see in front of you, right? Like the animal has legs that don't function as well or whatever, right? Because they're old-looking legs. But why is it relevant that it was accelerated in and of itself? This is maybe a bit of a tangent from your talk. I know it's a broader issue. But I'm curious, why do we think at all that the gap is relevant as opposed to the actual age of the brain?
Well, as Doug was explaining, actually, the age of the brain itself doesn't give us more information. I mean, it's like we just know like the age of the people, that we just know that, for example, someone is 60 or someone is 50, but from their age, we cannot know how well they perform, for example. And this brain age gap give us some, more information about how their brain is aging or working, like it's 5 years older than it should be, for example.
Well, I guess because if I'm just taking two people, right, one is 30 and one is 70, I say, well, I bet you that the 30-year-old is going to perform better on this cognitive test than the 70-year-old, right? And I'm probably going to be right. And why am I going to be right? Well, because their brain is older, right? Their brain is older, therefore they probably are not going to perform as well because it's older because of some atrophy of gray matter or whatever that is, right? But maybe the 30-year-old is a little premature, so their brain really looks like they're 40, and maybe the 70-year-old is like preventative, right? So maybe the brain really looks 60, right? So in that sense, I know that they're highly correlated, but there's still some differences, right? In the sense that, well, actually the gap between the 30-year-old and the 70-year-old would be smaller if the other person had accelerated aging and the other person had decelerated aging, right? And then that would be, so I always thought that the brain age was sort of like, that's just a slightly better version of age, right? We all know age is relevant because your brain declines. But some people decline a little bit less or a little bit more, and shouldn't that be the relevant factor as opposed to whether it was itself accelerated or not? Does that make sense?
Well, I'm going to let you want me to? So I'm not the presenter. What I would say is that the issue is not between a 30-year-old and a 70-year-old. The issue is between two 70-year-olds, right? So why does one 70-year-old do better than the other 70-year-old? One explanation might be that one of the two has an accelerated brain age and the other doesn't.
No, I think it's that one has a younger age and one has an older age, right? So it is important that a 30-year-old is better than a 7-year-old, right? That's why we're investigating this whole phenomenon to begin with. And why would a 30-year-old be better? Because we think their brain is younger, right? And that's exactly the phenomenon we're talking about, brain age.
Brain age is just a term for brain health. Right. It doesn't actually mean the brain is older. It just means one brain is healthy.
So brain health is what matters. Exactly. Right. But I think acceleration or deceleration is really a separate point than the brain health itself. Right? If you're both, if two people have the same brain health and one person was more accelerating getting there and one was not, we still don't necessarily expect those people to be different because what we're saying is your health is what matters. Right? So it's not the gap.
But
you see my point?
Would you agree that, for example, with, let's say we are not looking at brain age, we are looking at gray matter volume or gray matter thickness, then do you agree that, for example, someone with lower gray matter volume has worse brain health, right? Or with lower gray matter thickness?
Yeah.
So then for estimating brain age, we use that metrics as well, right? It's not something else. It's based on like the gray matter volume, gray matter thickness, or the structural changes in the brain. So again, I think that accelerated aging or decelerated aging can give us some information about the brain health, right? Because it uses the brain structure to estimate that.
Potentially, yeah. I would be curious to know what's really going on under the hood there, right? I think there's some presuppositions built in, but yeah, I don't want to belabor it on this point, but we can talk later.
Can I just follow up on that? Did you check if there's a lot of brain age gap in younger participants? Because just kind of, related to William's question, maybe if someone is 20 years old and they have a brain that looks like 30 years old, is that really, aging, or is it just some kind of inaccuracy determining brain age? I was just kind of wondering if there were big brain age gaps in younger participants and what it meant.
You mean if there is a higher brain age gap in younger participants, then it affects like the analysis that we are seeing the relationship
Just
with brain age gap?
if you check that. Or,
Yeah, that would be great to check actually. I haven't checked specifically in this study. I mean, we've been using brain age gap measure a lot during the past years, and with some other studies, I remember that, for example, sometimes it was the case that for a 30-year-old, as William said, that, for example, they were a little bit more mature and they had like 40 years or 44 as their estimated brain age. I haven't checked in this sample specifically, but it would be good to check that. And also, I think then it is the case, we can separate the analysis between like the younger versus for older adults actually in our sample.
All right, I think on that note, thank you very much for your talk.
Thank you.
Great.